Good afternoon, everybody. I'm Sara Ashpole, and I will be your chair for the Species at Risk session. We have 4 extraordinary lineups, including 1 student presentation for the judges coming in at 3 o'clock. So I'd like to get started. You will have about a 10-minute time period for your presentation, 10 to 12 minutes, and then a few minutes for questions. I will give you the signal at 10 minutes to wrap it up. So our very first talk, we're going to welcome Meredith, Status and Conservation of Suburban Population of Jefferson Salamanders.
Hello, everyone. I'm a postdoc at the University of Toronto, Mississauga. I'm studying with Dr. Rosalind Murray at the university, and I'm going to be presenting some results from our first field season working with a population of Jefferson salamanders in the Credit River Valley. I'm just going to briefly mention what a Jefferson salamander is. Many of you may probably already know. They're mole salamanders in the family Ambystomatidae. They spend most of their life underground, and they're one of the earliest spring breeding amphibians. So often they're coming out even as there's snow on the ground. Our population last year came out on the 28th of February in Southern Ontario, so they come out quite early. They typically breed in vernal ponds that are dry for part of the year. And everywhere that Jefferson salamanders occur in Canada, they co-occur with unisexual salamanders. The unisexual salamander complex is a complex of all female hybrid species that exhibit kleptogenesis. So they steal spermatophores from a host species. It's highly complicated. You could do an entire symposium on them. In fact, Société for the Study of Amphibians and Reptiles just did in June. And I'm not going to go into all the nitty-gritty on unisexuals. I'm just going to focus on, for our Jefferson-dependent ones, they're primarily blue-spotted Jefferson hybrids, typically with 1 copy of the blue-spotted genome and 2 or more copies of the Jefferson salamander. In Canada, Jefferson salamanders are on the edge of their global range. So they only occur in Ontario, as far as we know, and their populations there are very isolated. If you look on the right, you can see the red squares also represent areas where populations have not been seen since 1998. So there's also been a lot of extirpation. Many populations are occurring in these highly urbanized areas. So this is definitely a peripheral population. And in fact, they have experienced a 90% decline over the last 3 decades. And Jefferson salamanders were listed as endangered in 2011. The unisexuals were listed as endangered in 2017, so they have been under protection for quite a few years. Our population in the Credit River Valley in Mississauga is, of course, right in the middle of all of this urban area. So we're right on the edge of the Greater Toronto area, where Toronto's one of North America's largest cities. And this area, since 1967, has experienced rapid land cover conversion from agriculture into residential and commercial developments. And it was also last studied in the 1970s, right around when that conversion was just starting to happen. So right around when we started to see agricultural fields changing into neighborhoods and cities. And since then, there's not been a lot of research on this population. So one of the primary goals that we had starting out the study was to assess the breeding adult numbers, see how many are there now that all of that urbanization has occurred in the Mississauga area. We've had this basically almost entire changeover from agriculture and rural areas into more residential areas, and also identify what are the current threats to these populations at this site, with the goal of informing conservation and habitat remediation at the site. So to do this, we mimic the methods that were used in the 1970s. So Wayne Weller in 1980 set up drift fences around the pond that he studied. So we used drift fences and pitfall traps as well. One around an original breeding pond where they were known to be already, and then one around a novel breeding pond where they were recently discovered in the last couple of years to be breeding. The new pond was a man-made pond, and we were unable to trench the drift fences in. So we actually employed a novel method of using sandbags to create that ground seal around the drift fence and box funnel traps on the outside to capture salamanders. We weren't sure if it was going to work. Turned out it worked pretty well. So we had just as many, if not more, of the salamanders that were captured and marked going in were marked coming out. So we had a similar proportion of ones that were captured coming out that had been marked going in. Yeah, that's the way to phrase it. And we sampled, surveyed these every 12 hours, basically from the beginning of March all the way through late April. For every salamander that we captured, we recorded which pond it was found at and the trap location, which lets us look at their general direction of movement. We recorded length, mass, age, sex, and for a subset of the salamanders, we took tail tips for DNA so that we can determine ploidy and the proportion of unisexuals in this population. Every salamander also received a visible implant elastomer mark. So there's 6 possible positions, 4 possible colors. They had between 3 and 5 marks to allow us to be able to identify all of our individuals. And we took photographs of every salamander as well. As far as assessing the threats in the area, we looked at a number of water quality variables, and we assessed pond depth using the highly scientific meter stick, assessed predation with camera traps and visual observations of any carcasses observed. For road mortality, we looked at their movement direction relative to nearby infrastructure. And also one of our team members, a graduate student at the university, Sara, is developing or has developed a citizen science app. So people in the area will record when they see animals on roads. And then I also have an undergraduate currently looking at invertebrate communities in the ponds, and we're hoping to relate that to prey availability for these salamanders as well. Preliminarily, we marked 693 Jefferson salamanders in our first spring. As compared to the 1970s, it seems like we have pretty similar numbers since the captures per year in Weller 1980 were up to 624. We have found a significantly biased sex ratio towards females. Actually, if you look at the y-axis on this graph and notice where it's starting, it was significantly biased in the 1970s. So a balanced sex ratio would be 0.5, and it was significantly biased from 0.5 in the 1970s, and now it's even more significantly skewed because our results were significantly skewed from the 1970s. So it went from 11.4 females to every male to 167 females per male. We don't have the genetic data yet, but this suggests that there's a very high proportion of those all-female unisexuals. Some preliminary looks at movements in direction relative to infrastructure, roads, and buildings. At our natural pond, most of the movements were actually along these drainage channels, not very many relatively going towards more urban areas or from more urban areas. But at our man-made pond, we did have some salamander movement coming in from these more urbanized areas, although most of the leaving direction was not. It's likely that we have more movement in relation to that infrastructure because this population or this particular pond is in a slightly more developed area. So there's less option for overwintering or foraging habitat for these salamanders. So they may be having to make some movements towards more roads and parking lots. As far as the threat assessments, we identified a few relatively high threats. So one was pond hydrology. We noticed that the pond levels dropped to somewhat alarming levels at a few critical points in development. So they dropped fairly low during egg development so that we had some egg masses exposed. And they dropped during larval development to a point where we were worried that we were going to have increased larval mortality. So it is most likely an issue here that the ponds could end up drying too early during larval development. And then we observed some predation here. So we had some observations of predation. There were certainly predators present around, both egg and adult and metamorph predators. And we observed road mortality in other salamander species at the site. And we also observed at least one individual crossing a parking lot. So there is a risk there that they could encounter roads. We found 2 additional threats that we didn't anticipate. One was pet collection, whether intentional or unintentional. And the other was overpopulation by unisexuals. So we had that highly biased sex ratio. So there is an idea that over time, very isolated populations of the host species for unisexuals could become driven to extirpation by these all-female unisexuals. So eventually what was hopefully once a balanced sex ratio, you get more and more females. The unisexuals outcompete the diploids, fewer males relative to females, and eventually you have only females. And this has seemingly actually happened to some populations in Ontario. There are some Jefferson salamander populations in Ontario that are entirely unisexual. We have a lot of directions still to go, tons more work to be done. This is a massive project and we're just getting started. I just found out that we may have another 3 years. So I'm just going to highlight quickly a couple of things some of you may have seen Chris Botch's talk earlier about our visual identification, building a neural network model. And then moving forward in the next 3 years, we're thinking about doing some work with population genetics and connectivity of the populations in Southern Ontario, especially the ones in the Craig River Valley. Full disclosure, I'm not a geneticist. So if anyone has any recommendations or insights into how to approach these problems, I would really appreciate that. And we always need volunteers. And like I said, this is a huge project and collaborators are appreciated. If anyone's interested, we've got tons of data and there's more salamander species than I had time to discuss there as well. And with that, I would like to thank so many people. I can't even name all of them, especially grateful to the people who came out on long, cold, very, very rainy nights to help make this project happen. Thank you all.
We have 2 minutes if there's any questions for Meredith.
Great talk. Thanks so much. Is that working? Oh, yeah. Yeah, great talk. Thanks so much. That was really cool about the pet collection ones. How did you know that that was actually happening? Did you see people collecting them?
Yeah, so we didn't see them collecting Jefferson salamanders specifically, but we did see some kids with dip nets netting in probably they were netting for frogs, but I was like, you know, there's no way we could know that they wouldn't accidentally scoop up a salamander larva in there and just think, ooh, cool, and take it home. So that's why I said intentionally or unintentionally, because I'm not sure they're intentionally taking them, but it is a risk there. Yeah.
Anything else? Okay, thank you very much. Oh, sorry, is there one more? My apologies.
Do you think in a more great presentation? Do you think in a more natural environment that unisexuals would be less of a threat to a population for like extirpation?
Yeah, that's a good question. Actually, so the idea behind it is that in connected, when these populations are highly connected, you get more of kind of a source sink dynamic where the unisexuals overpopulate, but then the diploids get another pond that they can move to, and then they repopulate, and then the unisexuals go find them there, and then they move. So if there's more connected patches of ponds, it's not believed to be a problem, but because these populations in Ontario are so isolated, it is, yeah, it's a concern. Yeah.
Great, thank you, Meredith.
Oh, sorry.
Okay, I'd like to welcome our second speaker, Briar Hunter. Will be speaking about where 25 years of species management have left us. We only have 280 frogs and counting, Briar? That's what's left in the world? Oh, Jesus.
Not in the world.
Not in Canada? Okay, we'll find out.
All right. Yeah, just give the whole thing away, Sarah. It's fine. It's in the title. It's in the title. Yeah, I gave it away. Just spoilers. All right. Yeah, thanks. Welcome. Hey, everyone. So this beautiful frog here is the Oregon spotted frog. They are considered one of the most endangered amphibians in Canada, and they have a pretty limited Canadian range, which is only in BC, and within BC is within the South Coast region, which is where I currently work as an ecosystems biologist for the province. So I have the privilege of kind of coordinating and managing their recovery program. So I just want to start, before we get any further, with giving an acknowledgement. I work and play on the unceded traditional territories of the Coast Salish peoples, and two of our Oregon spotted frog populations in particular find refuge on the lands of the Société First Nation and Seabird Island Band, and for their stewardship of the land, myself and the frogs will be forever grateful. So, I don't know if you all get this or if it's just me, but a lot of people in my life know that I work with frogs. They know I specialize in frogs, and if you'll excuse my language, they know I am ardently obsessed with frogs. And so I get the question a lot, Briar, how are the frogs? And I used to say, good, good, and I'd show them cute pictures and try to endear them to the frogs. But recently, I've started to just answer honestly, and I just say, bad. They are honestly just doing bad. They are highly endangered. And so then this cues the questions, well, why are they so endangered? How did they get to this state? How did it happen? Well, as I said, they've never had a wide range. They're kind of restricted to the West Coast. You can go all the way down the West Coast here. And if you see the darker red, see if this pops up. The darker red is what the present distribution was in 2012, with the lighter being what it was considered historically, their range. So never huge, but there was an emergency assessment back in the 1990s, which determined that they had disappeared from up to 90% of their range. And so after this emergency assessment, they were designated as endangered in 1999. And this 2012 distribution was based on 4 known populations. There was only 4 populations left in Canada at the time. And since then, one of those populations is now presumed extirpated. We found 3 more, and one of those is now presumed extirpated. So it's a roller coaster, but still endangered. And so obviously this brings some implications. We have some work to do, and we're trying to reduce this, but it's hard to work with only 4 populations. So as my title gave away already, our current estimates are 280 mature adults in the wild. So how we get these numbers are we do egg mass surveys every year at all of our known locations, some historic locations, and then sporadically we try to go out and check areas that seem suitable. And it's been determined that just multiplying the number of egg masses by 2 gives us a pretty accurate estimate of mature population size. So this is how we get our population estimates. I will note that the effort has been more standardized on this since 2011. So some of the numbers there previous to that may not be consistent, but I'm also going to add some key points here. So I said that one of our populations was extirpated. We had another one, which is one of our largest kind of primary sites, discovered a couple years later. And then those 3 that were discovered in 2015. And I will make a note, these 3 that were discovered, they live in ditches. Who would have figured? The reason we found them was that people could identify them in fish salvages or out on the landscape, which is a really hard thing also with our Oregon spotted frogs, identifying some of those early developmental stages. So even having people on the landscape being able to identify these species has been difficult and has led to finding those 3 populations. So this just kind of gives you some more context for this fluctuation. And again, this is for all of Canada. This is combining all of them. And I'll also note, as you see this little asterisk at the bottom, we discovered this one population in 2015. We've only found egg masses there once. We've had a hard time finding any since then. So they could be there still, they might be gone. Really cryptic species. So just to break this down a little bit more, these are our individual populations. So you can see that the individual populations are fluctuating as well. The past couple of years, we've seen some dramatic dips, especially in that Morris population, which until now was kind of our largest, most stable population. Not great news. The worst news is that we don't know why. There's obviously lots of threats. Amphibians face lots of threats in general, but we're not sure what's causing these specific declines. So this is also just unfortunate because in 2014, Kissel et al. Determined that it takes 150 breeding females, which would give you a population size of 300, is the minimum you need for a stable population. And that's to bring your extinction risk below 20%. If you recall the numbers, Morris has only hit this benchmark 4 times since we've been monitoring it, and Mariah once. None of the other populations have ever hit what we consider stable. But we're trying to fight against this. Since 2010, we've released over 63,000 Oregon spotted frogs. This is in embryo stage, tadpole stage, metamorph. We've been trying to put them back out onto the landscape. And we get a lot of those from our conservation breeding populations that we have going. So most of that augmentation happens within the Mariah Slough system. So Chaplin is our main reintroduction site. And so I've tried to break it down a little bit here because we can say there's 63,000 thrown into the landscape, but it's not affecting all the populations since they're pretty isolated. So if you look at the blue dotted line, these are the frogs that we're putting back onto the landscape. But just a note that it takes at least 2 years for an individual to mature, so you shouldn't be seeing them impacting the population number for a couple of years. And we're not really seeing any impacts of this. We have really low retention. We're not sure why. So we're putting them on the landscape and we don't know where they go. They may not be surviving. They may just be leaving. We're putting them out there. We also have had huge reproductive success increases at the Greater Vancouver Zoo since 2020. That's why we have a really nice big spike there, and we're hoping that we'll start to see the impacts of that. That would be starting now or the next couple of years. You can also see that Chaplin has just started getting wild egg masses. So as I said, this is our one reintroduction site, and it just has a few years there because we found our first wild egg masses there in 2020. And we've had between 1 and 6 egg masses there ever since. So there's a few frogs hanging around. Where have the other 60,000 gone? We don't know. So I'll just give you a little bit more context for Chaplin. As I said, Chaplin Road wetlands are within the Mariah Slough system. They were created in 2009, 2010. You can see kind of at the top of the image there, that's our main, what we would call our Mariah Slough population. And then further down is this created Chaplin Road area. If you come out on Tuesday, you might get to see Chaplin. And we're assuming that they're separate populations based on the distance. The frogs don't typically travel that much. There's a possibility they're traveling between, but low chances of any interbreeding between them. Our first translocations bringing zoo-bred frogs into this Chaplin occurred in 2011. And as I said, we got our first wild egg masses in 2020. That's exciting. Don't know why it took 9 years, but we have them now, so we're just trying to keep them there. One thing we've tried to do is we have these wetlands, we've been putting tadpoles there, so why aren't they staying? Where are they going? So we've been looking into what are the potential limiting factors here? Why aren't they staying? And there could be a whole host of things. I don't know how many of you are doing this applied management on the ground, but you're just constantly making lists of what are your potential threats? What are your potential limiting factors? And there's so many. And how to decide which ones to tackle is like a year's worth of just brain thinking in general. So one that we highlighted was the habitat. We know where they are in the breeding season, but outside of that, for a lot of our populations, we just have no idea. And for juveniles especially, we don't really know what habitat uses they have, what habitat needs. So hopefully some of you got to go to Jen's poster yesterday, but Jen works at the Fraser Valley Conservancy and she's done some really hands-on juvenile and tadpole dispersal, trying to look at their habitat needs, habitat uses. And the objective, again, as I said, was to even figure out what they need. We know that we created Chaplin targeting for those breeding areas for our adults, but do we have the habitat that the juveniles need, that the tadpoles need? We're still not 100% sure. Still trying to figure it out. It's very difficult to trap and ID our juveniles and tadpoles. The Conservancy also recently came up with an identification guide for our early stages between northern red-legged frogs and Oregon spotted frogs. So that's hopefully going to make this a lot easier. But again, it's baby steps and you're just building on each other to try to use these tools and then answer the questions, which inevitably just bring up more questions. So as I've said, so many threats, limiting factors. I could go through all the threats, very similar to many of the other amphibians. I'm sure you're all aware. Let's just narrow down to one of these. Again, thinking through how we actually manage this. You can write bullfrog as a threat on a list. What does that actually look like? How do we actually figure out how to manage it? Are they directly predating on our frogs? We know we've seen that in the States. we're not 100% sure if they're predating on spotties up here in Canada. Are they competing for resources? Is it just there's not enough resources to feed both species? Is it, how do we manage them? Do we have to directly remove them? Is there a threshold that we can get them to? Or do we have to stop them from getting in at all, which is very difficult on our landscape. I don't know if you've noticed here in BC, they're going everywhere. And how many? Again, that threshold, can we keep them to a certain number so Oregon spotted frogs can compete with them? And how do we feel about removing, potentially euthanizing a species that's native elsewhere in Canada, but it's non-native here? We can get a lot of public pushback from that as well. I will say we have done some bullfrog management. We do go out and try to remove a few to try to keep it to a threshold, but we're still trying to tackle a lot of those questions. We don't know exactly how they're impacting each other. So ongoing. And just one final kind of threat challenge that I want to highlight is this public perception. A lot of people don't even know Oregon spotted frogs exist, let alone know how to identify them or know what the threats they are that they're facing. So how do we kind of raise this more in the public eye? How do we get people engaged? Very difficult, but hopefully this is a start. More of you are going to be aware of them. So this is sort of a call. We have a lot of data at the province. We've been doing monitoring for quite a few years, but we need more students. We need more researchers, people to collaborate with who are interested in some of these questions. If you're interested in bullfrogs, I can give you so many bullfrogs. Come and work with us. So here's maybe just giving that more specifics. As I said, we have a ton of data at the province and I don't have time to analyze it all. I have so many questions I could give you. If you have questions, I would love to try to give you some data to look into it. And yeah, whether it's you want to look at bullfrogs, you want to look at habitat, we have a lot of things that we could tackle and people with so much knowledge here within the province that would love to work with you. So yeah, with that, ton of people that we're working with, ton of organizations. Come find any of us.
Thank you, Briar. We actually do have time for a question.
Oh my God.
Any questions for Briar?
It's a first.
Yes, we do have one.
Hey, Briar. So I had heard about those populations that were found in ditches. Alicia Switzer told me about that a couple years ago, and it was exciting, but also I think that there's a perception even amongst the biology community that Oregon spotted frogs, they're over by Aldergrove and at Mariah, and I'm a consultant, so I think that we need to like spread awareness that they could be in ditches, maybe even in Surrey, so that there's awareness to be looking out for them. Yeah, I think that that's one thing that we could do right now.
Yeah. 100%. That's that public perception piece, right? Even people who know about them think that they're further out near Agassiz or they're those big historic populations, but we had no idea they were in those ditch systems, and we're still unsure of the connectivity between some of those ditch systems because we don't know where they are for 75% of the year. So we assume that they're within their restricted watersheds, but yeah, tell all your friends, check your ditches. If you have photos, take photos of everything. That's something Jen and I have been pushing, especially with this identification tool that we're just trying to put out there, is if you aren't sure, take like a bajillion photos and send it to Jen or I, and we will work to identify it because sometimes we can't even tell.
Great. Thank you very much.
That was
Thanks, Briar. Yeah, they might be a little bit more mobile than people realized. Yeah, not you. I'm talking about the frog. Our next speaker is a student for the judges in the room, please. So I'd like to welcome Kara Atkinson for Small Mammal Snacks. Great start to a title. Long-term trends and risk factors for prey communities of at-risk snakes in BC's interior grasslands.
Hi, thank you. I'm very honored to be here. So yeah, I'm a grad student in Carl Larson's Army, I guess, and I'm also co-supervised by Dr. Leanne Isaac. And apparently I should switch careers and be an artist because I just couldn't, I wanted to find something that would represent this. So Yeah, to be clear, today, while we all love and admire all of the wonderful snake species we have here in BC, today I'm going to be talking about this. So for a bit of background, in Canada, a lot of the snake species tend to eat small mammals and insects. That's the majority of the prey base for a lot of these species. And if you didn't know, rodents tend to have really strong population cycles. This has been tracked all over the world. You see they have these really high highs, and a few years later, they have these really low lows. So there's these really huge population fluctuations that we can expect. So this has a few impacts on snakes. Snakes that are born in high prey years tend to be larger throughout their entire life and tend to have better fitness, which we'll see in a moment. Snake body condition has been positively associated with prey numbers. So again, they have better body condition in those years where you have those prey peaks. This has been shown in a study in Australia by Madsen and Shine. And again, body condition in prey also influences snake reproduction, and both of those things have been shown to be positively associated. So you have better snake reproduction when they're in higher body condition and when there's higher prey years. Makes sense, right? So there's another figure from that study. So what I'm going to talk to you today about this project. So we're here in Kamloops, BC, which you guys have heard a lot about from Carl's talk and some of the other students in this lab. So like a lot of the other interior grassland projects, we have a very dry, arid environment. There's lots of really cool species that live in this area because it's a really small part of the province, but it has a huge biodiversity. It's also very threatened, as you might have seen from a lot of the agricultural influxes on a lot of the photos in other people's presentations, so I won't go into that. So there are about 5 main snake species. We have a few garter snakes, but I kind of didn't have a lot of room on the slide, so I stopped myself here. But yeah, we've got rubber boas and racers and gopher snakes and rattlesnakes and garter snakes. And they prey on 2 main prey species in the grasslands. We don't have a huge diversity of the small mammal population. So when we're going out and trapping, a lot of them are one of these 2 individuals, and then they eat insects, seeds, and graminar vegetation or grasses. Something that I learned when I started this project was that people actually say like, oh, the mouse will predate on seeds, which I've never heard, but anyway, I digress. We also have a lot of other endangered species that live in this area that also prey on them. So while I'm not talking about them in this talk, we also have like American badgers and burrowing owls, and all of these rely really heavily on these prey populations. So this project, we have trapping data from 1997 to the present, which Carl has really meticulously had a project, like kept this long-running project to keep really good data for this. So I had the honor of working on that. So basically, we track these small mammals, we take our measurements, which they are not always impressed by. We have 3 consistent grids, so we're going to the same area all the time. So we have a lot of consistency throughout the project, which is really great for a project that's been running for almost 30 years. And then I've taken climate data from the area. I got this from Climate BC, which has a really good combination of like satellite imagery and Environment Canada. So it's a really great data set. And I had some objectives that I'm going to go through in this talk. So I want to talk about what climate variables affect small mammal abundance. Are there any trends present? And what can we expect in the future? And I kind of gave it away a moment ago, but I am going to be talking about climate change. You might have thought you were escaping from that by being in this room rather than the climate change room, but sorry. So we had some difficulties. We had stubborn mice, we had angry mice, and they do break the skin a lot, actually. And we had black widows in a lot of our traps. In one of our grids, when I was cleaning them, about 60% of the traps had a black widow in them. So lots of little surprises there. Carl had warned me that there would be sometimes neonate rattlesnakes in the traps, but that was a welcome surprise. So some of the results I have, just to orient, the top we have the montane vole, and the bottom we have the western deer mouse. You can see that we have those really the inherent peaks and lows of these populations. And the deer mouse is really ubiquitous across the landscape. So we see them basically everywhere. All of those predators will have eaten deer mouse in their life. The voles, on the other hand, you get them in sun sites. Some years you have 0 voles. Sometimes you have a whole bunch. So they're really not a reliable prey population in the way that the deer mice are. I also wanted to bring your attention to, even within the same year, these sites, some of them are less than a kilometer apart, and you can have up to like a 40 animal difference in that area. So when we're doing things like salvages and moving the animals away and they return to the same area, they might have a reason for that because there might actually be a really huge difference in prey availability between that really small area. So again, really, really strong differences. So yeah, going forward, I'm going to talk about the deer mouse just because of their ubiquity on the landscape and the potential for effects that they have on the snake populations. So I chose for like the 4 seasons for my temperature predictors, and I adjusted those for the most biologically relevant windows. So like if it was summer is June to August, but it was more biologically relevant to just look at July and August, then I shifted that. So my models ran that. And then I also looked at warm season precipitation and then snow. So what you can see here is that the summer precipitation and the snow had small but significant impacts. Some maybe from winter temperature, but what I really wanted to look at is the July and August temperature and the September-October temperature. So those had really strong negative impacts. When the summer was hot, we had a drop in population almost every time. This is just, I took this from the climate data and I was slicing it up a bunch of different ways. This shows a similar thing to what Carl's talk showed earlier with an increasing summer temperature. But so I've shifted this into every 25 years. So the past 25 years have been significantly hotter in the summer months than any other year in the past 75 years. So when you look at it on that average, even though this graph doesn't quite show the high highs and low lows of like what the dry environment might be, when you look at it on a large scale, it actually is a really big difference. So the mouse peaks. Remember when I talked earlier about how the snakes really rely on those small, high prey years for reproduction and other fitness traits. When you look at it across the scale, it's not significant, but you can see a slight downward trend. We'll see what happens in the future, but there is a possibility that those hot summers are creating a lower carrying capacity for for the prey populations in these snake habitats. Again, so this is where I plugged in my predictive model for what factors affect small mammals, and I put it in the future climate prediction data. And what you can see here is that those peaks go away. So what now? Do we sound the alarm? I took this video from the Société Snake Project, which I have been on. They're lovely snakes. So in summary, we have these really high variabilities between sites and between years. We have really hot summers, and those are associated with low prey numbers. We have summer temperature that has been increasing by a significant amount, and we can expect that to continue in the future. So potentially we may have lower prey numbers and a lower carrying capacity, which could affect the carrying capacity of snakes in this environment. I wanted to highlight that like deer mice are really ubiquitous. They're not just in the grasslands, they're everywhere. Even if they disappear from the grasslands, they would not be a species at risk if they're persisting in these other environments. But for the animals that live in the grasslands, that can be a really huge impact if their prey population just shifts somewhere else. So I wanted to talk about prey management and what we can do going forwards. So for a moment, I'm not going to talk about snakes. I'm going to bring in one of the other species. So there was a burrowing owl study where they were talking about diet. And I just have this quote: we recommend managing Western burrowing owl habitats for species, especially rodents such as pocket gophers and voles that do well in urban habitats. So as we can expect more habitat fragmentation, we want to preserve the species that are going to promote predator persistence in the environment. Currently, only one grassland small mammal in BC has a management plan. That's the Western harvest mouse. So this is just some of the things that I took from that management plan. But they recognize that the fluctuations increase the vulnerability of the species to disturbances when numbers are low in spring and early summer. So having those really high highs and really low lows, if something happens in a low year, you're a lot more likely to see a strong and long-lasting effect to that population, right? It's a risk factor for that. Additionally, they recognize climate change, including the possibility of more frequent wildfires and intense drought as an increasing future threat. So going forwards, like I said, we can potentially expect a lower carrying capacity for deer mice in grasslands, but this could potentially be an opportunity to fill ecological niche. So this is a study that was done that I found interesting, but that climbing mooring could increase biodiversity of small rodents. So they allow those generalists might leave the area, but they're reducing competition for some of those specialists that live in that area. So in BC, we have 2 threatened or endangered rodents here that I'm just going to highlight for a second. So we have Columbia Plateau pocket mouse, which was previously known as the Great Basin pocket mouse, if you've heard of it. And we have the Western harvest mouse, which is endangered. So potentially this could be an opportunity to manage for both prey populations and the predator populations that rely on them. And yeah, so I just wanted to end with a take home that, you know, when we're looking and honing in on a species at risk, we might not be getting the whole picture. Sometimes a holistic view could help highlight some of the other factors that affect these populations and that maybe we can keep in our minds when we're making management decisions. Thank you. This is my study site.
Thank you, Kara. We actually have time for a few questions.
So I'm wondering if you've thought about this in a more philosophical way, and if you've wondered to yourself if snakes are going to follow prey into different ecosystem types, like maybe they're going to move in forests where prey is more abundant.
Yeah, they definitely could. Which could give some of them an advantage, like Carl and Lindsay talked about how some snake species or some snake projects in this area have shown that the snakes that move up into those higher areas, the forested areas, do come back with better body condition. But like Lindsay showed, it also puts them at high risk for other things like wildfire. So while you're mitigating one risk by them moving into that better prey area, you might be opening up the risk for another factor. So it's really hard to make a generalization, I think, in that area, but that is definitely something that I thought about.
Has Carl done or planning to relate your data to some of those movement studies and actually look at the movement patterns on those low prey years versus high prey years?
Yeah, so unfortunately, my study site is on the south shore, and all the data we have was from the north shore. So there is no habitat, I guess, overlap between those two species. I have caught a number of gopher snakes on my sites, but I didn't collect data on them because I didn't have any permits. But yeah, we don't have any data on that.
Okay, thank you very much. Our final talk of the session is by Kyle, identifying Eastern Foxsnake critical habitat along the east coast of Georgian Bay at the Shawanaga First Nation. Well done. Okay, thank you.
Thank you very much. So as Sarah mentioned, my name is Kyle Vincent. I work as a biologist for Shawanaga First Nation. I'm here presenting today on behalf of the rest of our species at risk team. So Stephen Kell, Jay Derdinger, and Delaney Griffith. And yeah, we are fortunate to work on the traditional territory of Shawanaga First Nation, which also happens to be one of, if not the most herp diverse areas in the country. But today we'll be focusing on some of our proposed work and ongoing work with the Eastern Foxsnake we have pictured here. And I also just wanted to give a shout out. All of the great photos of snakes you'll see through this presentation were taken by Jay. Anything that you see that's a little bit blurry or out of focus was probably taken by me. So I just wanted to give them that shout out for some of the fantastic visuals. So just some background for those of you that aren't familiar with fox snakes. They are a large bodied snake, federally endangered. A large proportion of their global range is actually found in Ontario, but Canada, about 70% of that range. So conservation of this species in the province is very important. The Georgian Bay population, they're split into 2 different populations, which I'll show in a minute, has a strong fidelity to shorelines and islands, typically found within about 500 meters to 1 kilometer from the shoreline. So that's what you'll find in most of the literature that you see out there, and both the provincial and federal recovery strategies. But as I go into our work, we'll find that we're having some findings that kind of contradict those rough guidelines when looking for these snakes. The snakes in the Georgian Bay population can also be highly aquatic, sometimes swimming over 10 kilometers between mainland and island sites for different habitat uses. So this is the kind of population range that I was talking about. So you can see up in this area along Georgian Bay, that's the area that we're talking about. And Shawanaga First Nation is located right under that big orange star. And it's separated from the southwestern population by about 200, 250 kilometers. this population tends to be much more inland, less aquatic, which is really interesting. And so this is Shawanaga First Nation. It's separated into 3 main reserve parcels. The main one located here, which is where most people live, Shawanaga Landing down on the shore, and then Nayscoot, which is typically used a little bit more for like hunting and recreation. There's a little less human development up in this area. And so Eastern Foxsnakes at Shawanaga. Most of the initial observations that led to the start of this project came from community members who reported a wide variety of foxsnake observations, most of them concentrated in one area, which also happens to have kind of the most human development and habitation, but also observations from up to 7 kilometers inland, which is quite far outside of what you find in those recovery strategies that I mentioned in the literature. So that led us to take an approach with our species at risk program to more actively monitor these populations, doing more systematic surveys, and that started back in 2019. And so through those surveys and community observations, they've provided a ton of information on seasonal habitat, some population demography, but the locations of the critical habitat, which is essential for the persistence of this species, is still poorly understood in the area. And so that's what led us to kind of start this project up. And so our overarching objectives, because these snakes are still faced with things like road mortality, that's kind of the main one that we see at Shawanaga, but also habitat loss and unfortunately some human persecution. Understanding where that critical habitat is on the landscape is going to be critical to conserving this more local population. And so our 3 primary objectives are to improve our overall understanding of Eastern Foxsnake occurrence, population, demography, and habitat use at Shawanaga First Nation, to deploy and monitor the use of artificial nesting structures to help kind of augment the habitat that may be available to them, and to start a radio tracking study on some individuals to identify those specific locations of things like overwintering sites, natural oviposition or nesting sites, and then also the movement corridors in between them, because that's where the snakes tend to get hit on roads. And so the kind of overarching methods we're using, the mark recapture, which started in 2019. So those are visual encounter surveys, collecting location and morphometric data of those snakes, and marking those individuals with pit tags. So that work has been ongoing since 2019. Artificial nesting structures. Our plan is to build kind of a whole array of them across the potential habitat and some of the island sites. To date, we've installed 2 on the mainland. We have 1 more planned for Shawanaga Island. So that work is also ongoing. And then the radio telemetry study, we had planned to start this year, but due to some hiccups, that's going to be starting in 2025. And we kind of have the overarching goal of radio tagging 6 males and 6 females with a combination of some surgical implants and then also testing some less invasive tape or glue methods that's a little bit shorter term for some of those snakes as well. And so, although we haven't started the radio telemetry part, we'll have to present on that at maybe the next CHS. We do have some really interesting nesting and overwintering observations that we've happened to find just incidentally or through community observations that I want to highlight because they're very, very interesting. So we have had observations of breeding pairs, juveniles, and neonates. Again, most of these are concentrated on that hotspot I was talking about earlier, where a lot of the community, again, lives. And interacts with the snakes more directly. So that confirms that there is natural recruitment happening and that obviously nesting sites must be close by if we're seeing neonates and juveniles. We also have a nesting site that was accidentally uncovered in 2020 during some road work. So the foxsnake actually nested between the asphalt and the top of a culvert. And when they were resurfacing the road, they uncovered that clutch. And we were able to recover that, that nest, incubate them successfully, and release them back out into the habitat. What's interesting about that observation is that it was about 2.5 kilometers from the shoreline. So again, that falls outside of that about 1 kilometer buffer, which is what's suggested in the recovery strategy. We also this year found 2 potential hibernation sites, which we were not expecting at all. We were hoping to find that with the radio telemetry work, but we were able to make this observation based on community reports. One community member in particular saw a snake crossing early in the season, like early May, late April, in repeated years. And so we went out and did some targeted surveys there and actually found a snake coming out of the ground, again, 2.6 kilometers from the Georgian Bay shoreline. And then we had another snake about a week later, 5.5 kilometers from the shoreline, which again is way up on the mainland. A lot of the snakes in the Georgian Bay population, based on previous radiotelemetry studies in the early 2000s, found that they overwinter on island sites, and then they wait for the Georgian Bay waters to warm up enough, they come back to the mainland. And to us, this suggests that that's probably not the case for this population. They are overwintering somewhere on the mainland, but we'll have to do a little more follow-up on that location just to confirm that in the coming years. And so I just included a photo here. These are some of the neonates from that recovered nest from the road culvert that were hatched out and released. And then this is a shot of, it was a young snake, that first one from 2.6 kilometers from the shoreline, which we actually saw coming out of the ground along a rock cut along a road. And so you'll see also an overarching theme with all these observations that they're almost always anthropogenic. Again, there must be some natural critical habitat sites out there, but this one's actually, it looks natural here, but it's actually a big rock pile from when they did that rock cut. They piled it up, and there's all kinds of complexity and fissures there. Same with the 5.6 kilometer observation. Again, it's right adjacent to a huge big rock pile of Canadian shield. So that's kind of our ongoing theory. We'll have to do a little more work to confirm that. Oh, there's the snake highlighted there if you didn't see it. So just to look a little bit more in depth at fox snake local range at Shawanaga, I've thrown up a map here of the shoreline, and I don't want to reveal any specific locations, but I just drew on a rough buffer for what that 500 meters that's suggested in some of Ron Brooks' students' papers from the early 2000s and what's included in the recovery strategy. So just a very narrow strip. That's what we expect for the Eastern Georgian Bay fox snake population. This 2.3 kilometer buffer was what was cited in the provincial recovery strategy for the farthest inland observation based on NHIC records. And so one of our, again, confirmed nesting site and confirmed hibernation site is actually farther inland than that farthest previous record based on what's in the recovery strategy. And then I've also included a 5.56 kilometer buffer there. Our second suspected hibernation site falls basically at the border of that buffer. So when you look at that on a landscape scale, and we think about things like human development, or again, even just conducting surveys, it's a much broader area that we'd have to survey to actually document this population. And we're not talking about just like a one-off individual. We actually have critical habitat falling within those areas. So again, I think a little more evidence that we need to think more about maybe revisiting those recovery strategies and what's included in there. On top of that, we also have community observations from as far about 7 kilometers inland. So again, we haven't been able to confirm that through photos or our own surveys, but it's quite likely that snakes are actually coming even further inland than what we've already documented. So we're hoping that that radiotelemetry study can kind of further uncover what's going on with this population. Artificial nesting habitats. So we have this ongoing as well. We were able to install 2 nest boxes near known fox snake observations, one at that hotspot, which falls within that 500 meter buffer where we'd expect to see snakes, and one very close to that confirmed road project. Again, we were thinking, okay, now we've ripped up the road, repaved it. We at least want to replace the potential nesting habitat for them there. And so we made up those boxes. They were using a 1 meter by 1 meter approximately wooden frame design with wire mesh. And then we filled them with equal parts straw, mulch, and local leaf litter. And so that's what they looked like when they were constructed. That one's kind of right along the shoreline. As you can see, the water in the background there. And they were installed in November of 2023. The one that was near the road site was unfortunately ransacked by a bear sometime in 2024. So all we learned from this one is that bears are stronger than zip ties. We used reclaimed wood, so when we put the whole thing together, the latch didn't line up. Again, we'll have to revisit that design. But that left us with one nest box, the one that's near the water, which we think probably had the best chance of actually being used. And so when we went back to revisit it, there was snake sheds all over it. I saw some neonate snake sheds. And so I was going to leave it an extra week before we went digging through it to actually confirm nests. But when I read through the abstracts of the presentations at CHS, I saw Ryan's talk and the results that came from that. And I figured, okay, we better go rip through this thing real quick, very carefully, but just see if snakes had actually used it and nested in it. And surprisingly, what we found is that they had. we had success in that one nest box. We found 38 eggshells from at least 2, but I think 3 clutches. All emerged from their eggshells seemingly successfully. And I just drew a quick little schematic there to show you kind of the relative location in the box. So that's a top-down view. So all clustered fairly close together. And then a front view. So you can see the material. That's kind of the beige in the background. That's where the nesting material and substrate actually settled down to. But you can see kind of, again, relative location of those clutches. So unfortunately, we didn't have temperature or moisture monitors in the nest box to kind of figure out if this is clustering because that one corner of the box happened to have, again, the best thermal gradient or moisture conditions. But that's something that we're going to implement in future years and probably install a whole bunch more of these boxes just so we can kind of figure out exactly why they're being used the way they are. So yeah, very exciting. So overall significance, community observations formed the foundation of this project and documented many of those inland occurrences and critical habitat use, or at least led us to finding the critical habitat that we have so far before doing systematic surveys. So I think that really highlights the value of both Indigenous, local knowledge, and citizen science projects and how they can help inform conservation initiatives like the one that we're undertaking. We have learned that artificial habitat can work. Again, if you saw Ryan's talk, you'll understand how significant this is for Eastern Foxsnakes. And it happened quite quickly. We weren't really expecting to see them being used, if at all, within the first year. So that was really surprising. And again, with that artificial nest box, especially having 2 or 3 clutches in it, it kind of leads us to believe that maybe the nesting habitat is limited in the area, which is also a bit surprising because relative to the rest of the population, especially the southwestern population, this is pretty pristine habitat surrounding our population. Identifying those movement corridors will help to inform effective mitigation like signage, decreased speed limits, and wildlife fencing potentially in the future. And then identifying foxsnake critical habitat, Shawanaga First Nation can inform land use planning. This is Shawanaga's main interest, ensuring that hibernation and nesting sites are not developed as the community continues to expand in the future. So lots of work to come, very exciting stuff. I'm looking forward to presenting on some more of our findings in the coming years. And that's all I have. Thank you.
Thank you, Kyle. We're a bit tight on time, but is there any questions? Okay, good. We'll take one question for you.
I was wondering if you know of an actual nesting site that was like a natural nesting site that was close to the artificial one, or if there was one anywhere in proximity?
We suspect that there were, we found, again, a neonate and some juveniles within about 100 meters of where we put that box. So that was why we put it there. We figured it had its best chance at being used. But no, we haven't actually confirmed a natural nesting site. Some suspicions, but we're hoping that if we can tag some of those snakes, then they'll lead us to where they're actually nesting.
It would be cool to see if they've moved from using the natural one to the nesting box, or if that's a different snake.
Exactly. Again, it's that question of, again, is it a limiting factor having nesting habitat, or did we just get super lucky putting it in the right place and happen to be the right conditions and the snakes found it? We're like, oh, this looks perfect. We'll just use this. But yeah, hopefully more to come in the future.
Great. Thank you.